I'm very excited to introduce our speaker, Dr. Michael Wells, today. He joins us from the UCLA Department of Human Genetics. We're going to hear about his exciting work, so I'll just say that he works with human stem cell models and does cool, exciting things that are relevant to the work of many people in this room. His PhD was from Duke. He did a postdoc at Harvard before joining us here, and he has some really cool, unique things on his CV that I just think that if you get the moment to chase him down in a hallway, you should ask him about. He's the creator and co-director of the COVID-19 National Scientist Volunteer Database. So he's really doing research that is both, so his own research is really interesting and engaged, but then he also is engaging in sort of broader scientific endeavors. And he makes frequent trips to Washington, D.C. to meet with members of Congress to discuss budgetary issues and remind them that we all benefit from a strong, adequately funded research environment. So we should all be very grateful for the work that Dr. Wells does on behalf of science writ large. So I was super inspired when I saw this, and I thought we should all be, engaged in these ways. And so without further ado, here is Dr. Wells to talk about his research today.
Thank you very much for that introduction, for the invitation, and for all of you taking the time to spend the next hour and a half or so with me. So my name is Michael Wells. I'm an assistant professor here at UCLA. And today we're going to talk about my exploration of human genetic and phenotypic diversity through cell villages. So my laboratory is very interested in the origins of human trait variation. So our species is characterized by an immense diversity in neurological and psychological traits, as well as risk for diseases that are associated with those different traits. So we think differently, we behave differently, we interact with our environment in different ways. The origins of these differences have been debated for millennia, but we're starting to more fully appreciate that for many traits and diseases, there's a certain level of innateness at play, meaning we're kind of just born this way. Now, this idea that this innateness is present has been supported by decades of research, specifically twin studies that have been able to calculate high heritability rates for such diseases as autism and schizophrenia, as well as traits like neuroticism and aggression. Genome-wide association studies have pinpointed specific loci that are associated with trait differences and risk for diseases. More recently, large-scale exome studies have nominated genes as being of particular interest for different risks and traits. Now, while we're doing a pretty good job of identifying the genetic signals that are associated with these traits and diseases, we're lagging pretty far behind in our ability to understand exactly how they are contributing to these traits. The function of these variants is still very much a question. So given that our lab is primarily interested in the brain and how the brain develops, we are very interested in how our genetic backgrounds, how our genetic variation influences neurodevelopment and associated diseases. We don't know, for example, how these genetic signals influence the ways in which neuroprogenitor cells proliferate and differentiate, or how postmitotic neurons migrate and form connections with each other. These are still very much mysteries in the field. Now, in order for us to really start to make progress in understanding these relationships, we have to cut across many different levels of biology. We need to understand how these genetic signals influence gene expression, signaling pathways, and various cellular phenotypes. We need to know how this in turn influences the way the brain is wired and how this ultimately leads to differences in our behaviors. Now, early in my career, I used mouse models to try to understand some of these questions. But as a result of becoming severely allergic to mice, I decided to change course for my postdoc and start using human stem cell-derived models of neural cells to investigate these questions. So for those of you who aren't aware of how this technology works, the basis of this approach, what we do is we take human pluripotent stem cells, whether they be of embryonic origin or of adult origin, in the form of these things called induced pluripotent stem cells, where we can take blood or hair from people and convert them to stem cells. Regardless of what you start with, we take these stem cells and we coerce them to become the many different cell types that populate the developing human brain. The question I was wondering as a postdoc was, can we use these human models in which we say make neuroprogenitors which can give rise to neurons and astrocytes and oligodendrocytes, can we use these for high throughput investigations into the relationships between genetic variation and disease-relevant neurodevelopmental phenotypes? So that's going to lead us to the topic of today's conversation, something we're calling a cell village. Now, Cell Village is our novel platform that allows us to connect alleles, molecules, and phenotypes all from a single dish of cells. And the way this works is we take cell lines from many different people, we call them donors, and we culture them all into the same dish for our analysis. In doing so, we can connect alleles to gene expression to cellular phenotypes, all again in a very small amount of cells and a small dish. That is what we're going to talk about today. We're going to talk about how we make these villages, the technical hurdles we had to overcome to get to this stage, and how we're actually using this village platform to answer questions about how our genetic variation influences our phenotypic variation. Okay, a brief little overview. We're going to start by talking about our model of neuroprogenitor cells. That's the thing we're focusing on. It's the key to early brain development. And I'll talk to you today about how we developed a new method for creating these cells in a very robust and fast way compared to standard approaches. Next, I'll actually give you I'll talk to you about how we construct villages and how we extract biological information from them using a couple of different bioinformatic tools. Then I'll give you a little vignette on how we actually use the village to answer questions about how human genetic variation influences differential susceptibility to a viral pathogen, in this case, Zika virus. I'll then give you a glimpse into the future on the ongoing cell village studies we're doing in my lab that focus on autism, lead exposure, as well as various other exposures. Okay, so let's get started. This is work that I did late in my postdoc in Kevin Eggen's group at Harvard Medical School, at Harvard University and the Broad Institute, along with members of Harvard Medical School and the Novartis Institute for Brain Research, I believe that's what it's called, biomedical research. Okay, here was the situation we found ourselves in. We really wanted to be able to study brain cells from many different people, but the problem was the methods for creating brain cells from stem cells were slow, laborious, and had a pretty low success rate from batch to batch. Now, those standard approaches are known as the embryonic body protocol, which takes anywhere from 16 to 50 days to go from stem cell to progenitor cell, as well as a dual SMAD inhibition protocol, which takes anywhere from 11 to 17 days. Now, these are great. They've been around for 10 to 20 years. They're great because you get these nice pure cultures when they work. The methods for making these cells is believed to mimic what happens in development in vivo. And these have been functionally validated, again, for 10 to 20 years now. A lot of labs use these systems. The problem is some of these require selection steps that are quite laborious. We're talking about having to handle each cell line, passage them, FACS sort them. They can be dependent on such things as how many cells you plated at the beginning. And if you didn't plate the right number of cells, you won't know for 3 weeks that you plated the wrong number of cells. And the biggest problem is there's considerable batch-to-batch and cell line-to-cell line variability. It'll take you a long time to optimize these protocols just for a single cell line. So to do this for dozens of cell lines like we do, it could be it was a major hurdle for trying to make these villages. So to overcome these limitations, we devised what we call stem cell-derived NGN2 accelerator progenitors, also known as SNAPs. This is a 2-day induction protocol to go from stem cells to neuroprogenitor cells. Now, the way this works is we take our stem cells and we introduce lentiviruses that encode an inducible version of the neurogenin-2 gene. Neurogenin-2 is a very powerful neuralizing transcription factor in most mammals. So what we're doing here is we're introducing a tool, a DNA construct, so that when we add a drug known as doxycycline, it turns on this construct, and then when we take doxycycline out of the system, that gene is no longer overexpressed. We have very tight control of the temporal aspects of this overexpression. So we introduce this into our stem cells. When we're ready to induce to our NPC cell type, we add doxycycline. We also add some small molecule patterning factors because NGN2 can make the cells into many different types of brain cells. What we're trying to do here is limit it to neuroprogenitor cells of the cortex, specifically the prefrontal cortex. So we're trying to make a very specific cell type here. So we're trying to minimize the variation that's introduced by having different cell types in our system. We do this for 2 days. We have some selection steps in which we just add a drug that kills off the cells that did not become the cell type that we wanted. And then from there, we can passage, expand, freeze, do whatever we want. Essentially, once we get this done for a given cell line, we can use those cells from the same vial over and over again for many, many passages. And we have some cells alive right now that we did this protocol on back in 2017, and we're still using it today from that same batch. So what evidence do we have that we're actually making the cell type that we care about here? There's a series of experiments we did. I'll go through this fairly quickly to validate the identity of these cells. First of all, we looked at protein markers. So nestin, PAX6, SOX1. As you can see, our cells express these canonical markers of neuroprogenitor cells at fairly high levels. In addition, these cells form these things they're called neural rosettes. So early in brain development, very, very early in brain development, these cell types will form a tube that we call the neural tube, okay? And you can actually see something like that in 2D cultures in which the cells form what looks like the rosette of a rose, right? So you can see some of those structures there. So again, these cells are behaving the way we expect them to. In addition, it doesn't matter if you're looking early in induction, after a passage, after 10 passages, we're showing here that our cultures are fairly pure, which is nice because if we want to put them all into the same dish from different donors, we want to make sure that they all have the same level of purity. Importantly, it doesn't matter what the starting density of your cells are. This protocol works every single time, which really separates it from the standard approach that people use, which is the dual SMAP protocol. In addition, we're showing at the transcriptional level that our cells express markers of anterior dorsal cells, so they are indeed the dorsal telencephalic NPCs that we predicted. We also validated this at the single cell level, in which we compared our cells to in vivo fetal brain cells. And when we map them on to the cell type that they most closely resemble, we're seeing that roughly almost 85% of our cells do map onto the fetal neuroprogenitor cell type as expected. And when we zoom in on that class of cells into different subclasses, we're seeing 90% actually map onto the earliest type of neuroprogenitor cell we can envision, which is again as expected because that is what you see during the earliest stages of the neural tube development. So we feel pretty confident that we are indeed generating cells that model the earliest brain cells that you see in development. Behaviorally, these cells are multipotent. They become the astrocytes and neurons. Again, this is expected if they are indeed NPCs. You can see that again, that depending on the culture conditions, we can push them towards glia or neurons. Again, a standard behavior that we expected. They are capable of self-renewal, meaning they can be multipotent and can proliferate at the same time. This is a key aspect of neural stem cells. And they can form these 3D aggregates known as neurospheres, which again is something that many have shown neuroprogenitor cells are capable of doing in vitro. Now, this is all nice, but it only matters if we can do this over and over and over again across many different cell lines. That's what we need in order to build a village. As you can see here, these numbers are slightly outdated because they don't include the 40 lines we've made recently in my lab that we haven't finished the QC on. But what we're showing here is that when we tried this protocol on 115 different cell lines, it worked in 96.5% of them on the first try. Okay, on the first try. Again, there's 40 lines we just made. I think 100% of them worked, but I haven't added that to here yet because we don't have that nice quantification quite yet. Importantly, we randomly selected 30 of those lines, and they all passed the multipotency assay, meaning they are behaving as NPCs. So this protocol is highly robust. We then confirmed that using single-cell RNA sequencing, where again we mapped our single-cell profiles onto in vivo fetal brain tissue data sets. And again, you can see a vast majority of our lines are mapping onto fetal NPCs. Okay, so at this point, I hope I've convinced you that our in vitro model of early brain cells is indeed valid, which leads us to the next phase of this presentation. How we actually use these to build a village, and what exactly is a village, and how do we get information from a village. This work, again, was done at Harvard in close collaboration with Steve McCarroll from Harvard Medical School. This was a true collaboration in which our lab was the wet lab, essentially, and the McCarroll lab was the dry lab. Over the course of 5 years, we developed this protocol, and really excited about what we were able to get out of it. Okay, so again, we're trying to understand how genetic variation influences cellular behaviors, right? Both at the transcriptional and cellular phenotypic level. Now, typically, if one were to try to do this, their incubator might look something like this, because for each line that they're trying to investigate, they'd be in a separate dish, because you can't mix cell lines. The problem with this is it introduces a considerable amount of well-to-well and run-to-run variation. That can actually mask your differences. So you have patient lines and control lines. You might not be able to see those differences because of this technical variation. That technical variation comes from things like cells in the corner well might behave very differently from cells in the center of a plate for no reason other than the fact of where they're located in the plate. So that's one issue. Secondly, this setup introduces many challenges in control and rigor when you're trying to process a lot of samples at the same time. If you were to try to understand how each of these lines may differ at the transcriptional level, you would have to prepare RNA sequencing, so bulk RNA sequencing libraries, from each of these different wells. And we are not robots. We are human. That will introduce a lot of variation just on the technical side, trying to prepare all those samples at the same time. In addition, this is very expensive to keep things alive like this for a very long time, and it can limit the number of hypotheses you can test because you're limited in your ability to perform these experiments. So with all this in mind, we asked, what if we just did this instead? What if we took all these lines and put them all into a single dish? And that's what we did, and that's what a village is. So here's how it works. We take dozens of different donor cell lines. Again, these are human pluripotent stem cells. And for each of these lines, we collect some genotypic data, whether it be whole genome sequencing or SNP array profiles. We're going to use this data later on because essentially what we do is we use their genome as the barcode. So when we're trying to figure out who's who at the end, it's their genome that is giving us that information. We then take each of these cell lines, we culture them separately for a brief period of time so we can induce them to the cell type of interest, in this case, neuroprogenitor cells. And then once each of these lines have passed QC and we've banked them and done everything we want to do with them, then we throw them together into a village. What we've been doing recently is expanding these lines, freezing them all down, and then when we are ready to make a village, we wake them up, wait a few days, and then pool them. So we're now at the point where we can go from frozen vials to village construction within about 5 days. Okay, so what we're doing here is we're capturing genetic, molecular, cellular phenotypic variation all in a single dish, but how do we actually get information back out? Well, there's two tools that empower us. One that's called dropulation, which is based on single-cell RNA sequencing of the cells in the village. And that not only gives us the full transcriptional profile of every cell that we sequence, but using this algorithm, we can actually identify the donor of origin for that given cell. So I'll show in a couple slides here how that allows us to do things like expression quantitative trait loci detection. We also use something called Census-seq, which helps us get cellular phenotypic information out of the village. Now, Census-seq is based on low coverage whole genome sequencing, costs like $60 a sample, it's very cheap. And all it's doing is exactly what its name implies. It's running a census on the village. Who's in the village? What percentage of the village do they represent? So if I had 100 lines and I counted them all really well, and I put them all into the village at equal contributions, well, census sequencing should tell me that each donor is 1% of the village. And depending on any sort of stressor we apply to them, or time, or fact sorting, that representation might change with those different conditions. And that allows us to see how the donors may differ in their phenotypes. And I'll give a couple examples of that as well here in a second. Okay, let's talk a little bit more about dropulation and how it works. What dropulation is doing is, again, it's based on single-cell RNA sequencing. What it's looking at are variable SNPs within the transcript. So it's using natural genetic variation across the donors as a barcode. And here's an example of what it might look like. The algorithm is scanning hundreds of variable genomic sites. And for a given cell, it's starting to calculate the likelihood that that cell came from one of these candidate donors. And eventually, it identifies a donor that stands out above the rest as being the most likely donor of origin for that sequence cell. And we run this algorithm across every single sequence cell in our village. And at the end, again, we get transcriptional profiles and donor of origin. And it's just by looking at the genome. We're not introducing anything into the system to help this out. So what this allows us to do is look at how genetic variation influences variation in gene expression. So what we typically call that is an expression quantitative trait loci, or EQTL. So if you're unaware of what that is, just look at the far right there. This is a toy example here. Essentially, donors that have the AA genotype at this theoretical site have low expression of this theoretical gene, whereas donors with a CC genotype at the same site can have high expression. When you see a significant relationship across genotypes, that's what we determine to be an EQTL. Now, an example of this that we recently published in our cell stem cell paper back in March involved a village of 44 different donors, okay? All of which we made into neuroprogenitor cells. When we did this, we found nearly 1,000 E genes, meaning we found about 1,000 genes in which we could identify an EQTL in them. What's particularly validating about this approach is that when we compared our set of E genes to E genes identified in fetal brain tissue, so actual in vivo tissue, we saw high concordance with what we found in vitro. So again, what we're finding in vitro seems to match up with what we see in vivo. And if there's any developmental biologists in the room, you also see that we've identified a series of E genes that play known roles in brain disorders as well as just typical brain development. And so from a single data set, we're essentially creating something that allows us to produce many different hypotheses that we can then track down later. Another problem this system solves has to do with standard GWAS studies, so genome-wide association studies. So these are very powerful tools. What they typically do is look at tens of thousands of different people. They look at their genomes and they look at certain traits. Okay? And so in this case, there's two GWASs we're talking about here. One is cortical surface area, what they got from brain imaging, and schizophrenia. And they're scanning the genome for sites that are associated with these traits or this disease in this case. But the problem is the loci they typically find are usually quite broad. They'll find a loci that encapsulates many genes. And so it's very difficult to fine map that loci and identify which gene or which variant is actually contributing to that risk or to that phenotype. What's nice about a village is that you can get that information at the same time. So in this case, one of our EQTLs was overlapped with this cortical surface area GWAS, but was able to pinpoint it to this very specific read-through transcript that is CRHR1, which is cortical tropin releasing hormone receptor 1. With schizophrenia, this paper that came out in Nature in 2014 found 108 different loci. But again, it was very difficult to pinpoint which genes were actually relevant to the disease. But in this case, we found overlap with this SRR gene, which is serine racemase, which plays a really important role in NMDA receptors, which is a critical component of brain development and brain circuitry. So again, we're providing evidence that's taking these in vivo GWASs and using it to fine map the actual genes that might be relevant to these diseases and symptoms. What else can we do? That's what we're getting from the transcriptional side. What about the cellular phenotypic side? Again, we turn to census-seq for this. Census-seq is using the same idea that Dropulation uses. It's using the genetic variation in our donors as a barcode. But in this case, all it's doing is it's taking a mixture of DNA, a bulk mixture of DNA, and inferring how each donor contributes to that DNA mixture. It's usually pretty quick. Within a few iterations, is able to figure this out. We start with the assumption that each donor is equally contributing, and then it's making these iterative changes so that its prediction for the mixture matches what's observed. Now, I think the most important question is how we can actually use it. There's a few different ways we can actually use this to look at cellular phenotypes. First of all, one of the most common ways we use this involves FACS sorting. Okay? So FACS sorting is where you can take cells, you can label them with different antibody markers. Let's say it's a marker for genes that are infected with the virus versus uninfected, or cells that are uninfected or infected. Or let's say it's cells that became neurons versus glia versus state NPCs. Whatever it may be, you can FACS sort your cell population into these different bins based on that phenotype. We can then run census sequencing on each of those bins. And what that allows us to do is calculate what we call an abundance ratio, or the donor's relative representation in the two villages. This can actually serve as a quantitative phenotype. So from a mixture of different donors, you can actually characterize their phenotypes all simultaneously, all in high throughput. Another thing you can do is just have time be your friend. You can just sample your village over the course of multiple different time points. So if they have differences in growth rates, you can pick that out. If, let's say, you're doing a model that is a cell death model, apoptosis, or looking at neurons and focusing on Alzheimer's, for example, you can actually see how the composition changes over time as a function of those different intrinsic behaviors. Now, I say intrinsic behaviors because some of the work we've done in our lab is trying to address this question about whether or not what you're seeing in a village is actually an autonomous behavior of those cells, or is it some weird thing that happens when you mix a bunch of different people together? Our brains develop with, for the most part, a single genome, right? There's not 45 different genomes in your brain as it's being developed. So we wanted to make sure that we were not creating some false environment that was creating all these data sets that really had no relevance to brain development. So some of the work we've done has looked at growth rates. So we took 12 different donors, we cultured them in a village, but we also simultaneously cultured them in an array. So we cultured them separately. We calculated growth rates for both. For the village, we used census-seq. For the array, we did our standard imaging-based growth assay. And as you can see, the concordance is very high. The correlation between the two different growth rate calculations are very high. You know, R is above 0.9. We also wanted to ask the question about whether or not villages are generating random data, or can you replicate the data you get from a village. So what we did here is we created the same village in two different waves. Wave 1 was done at time 0. Wave 2 was the exact same donors, but they were pooled a week later. And as you can see here, again, we're seeing a high correlation between the growth rates calculated using these two different approaches, or these two different waves. So what we're trying to communicate here is that the village is actually identifying or measuring cell intrinsic, cell autonomous behaviors, and it's not just generating a bunch of nonsense. Another paper came out about 2 or 3 months ago showing something very similar, in which they had a village of 3 donors, both in a culture and an array, but they looked at single cell gene expression analyses or data sets, and again found that what you're seeing in a village is pretty much what you'd see if you're culturing these things separately as well. Okay, let's get into the meat of the story here. How do we actually use this to answer important biological questions? So this is a little vignette on how we used this to identify how human genetic variation influences the susceptibility of these neuroprogenitor cell types to Zika virus infection. So I hope some of you remember Zika. It obviously fell out of the headlines because some other virus kind of took its place. But Zika is still considered an emerging global threat. Okay, this is a flavivirus. It's in the same family as dengue, West Nile, yellow fever, primarily spread through mosquitoes. There have been periodic outbreaks, the most famous of which took place in 2015 and 2016. It started in northern Brazil and actually made its way through Central America and into southern portions of the United States. Now, most people don't even know they're infected. About 80% are asymptomatic. Those that do show symptoms are quite mild, like fever and rash and conjunctivitis. But there is a particular population, a demographic that we are very concerned about, and those are pregnant women. Because if you're a woman and you're pregnant and you are infected with Zika virus, your baby is 17 times more likely to develop a severe neurological birth defect, the most famous of which is microcephaly, or small brain. This is what made this virus so famous back in 2015, when these babies were being born with very small heads and small brains in Brazil. Now, early work to try to identify which cell type this virus targets found that it preferentially targets proliferating progenitor cells of the developing brain. So with this in mind, we asked the question, can we actually infect our neuroprogenitor cells with Zika virus as our model, a good model for this infection? And perhaps more interesting to us is their variation across donors in their susceptibility to this infection. And as you can see, there's a huge chasm across our donors. So some of our donors, as you can see on the far left, are completely resistant to Zika virus, at least within the confines of our experiment. Others show nearly 100% infectivity rates. Now, these are cultured separately, but they're infected at the exact same time. So this is unlikely to be an issue with batch-to-batch variation for our virus or batch-to-batch variation for our donors. These are all done simultaneously. So this led to the question, if we're seeing this phenotypic variation, can we use a village to explain this phenotypic variation at the genetic or molecular level. So before we can get to that question, we needed more information about how Zika virus actually infects cells. What are the genes in the host cell that this virus manipulates in order to infect and replicate? So for that, we performed a genome-wide CRISPR-Cas9 Zika survival screen. Essentially what we did here is we had a library of CRISPR guides that we can introduce into our cells all at the same time, so that each cell presumably only has one gene mutated, okay? And if you do these in large batches, I had 36 giant plates of cells with these CRISPR reagents in them. Essentially, we got to the point where we can infect with either Zika or mock media. You wait a certain amount of time, you harvest, and all you're doing is you're trying to quantify the enrichment or depletion of those guides. So this is it's a 10-day experiment. We're looking for loss of function genes, and the hits in this case are those that are believed to be involved in Zika infection and cell death. So here's what this looks like. We found a series of genes that are called host factors. These are the genes that when you get rid of them, it makes the cells more protected from infection. Why? It's because you've removed a very important component of that cell that the virus needs to infect and replicate. So we're seeing many of the different gene classes that people have found for other flaviviruses like West Nile and dengue. These include heparin sulfates, which are important for how the virus attaches to the cell, integrins, which is how the virus actually gets into the cell, endocytosis, which is a way in which the virus actually not only enters the cells, but gets released into the cytoplasm and goes to the ER membrane complex where it replicates. So many of these genes are things we expected. A few others that we did not expect, like these integrins, help lead to the identification of how exactly the virus gets into neuroprogenitor cells. On the flip side, there are certain genes that when you get rid of them, you increase the risk of infection. Now these are called restriction factors. These are the genes that our cells have to protect us from these infections. So if you get rid of them, you're basically getting rid of your protection. These included genes like IFNAR1 and 2, STAT2, and a very interesting gene called IFIDM3, which you'll hear more about in this slide. What we did was we took the results of our Zika screen, which had about 125 genes that reached genome-wide significance. We took that 44 donor village I described earlier, in which we found all those different E genes, and all we did is cross-reference them. Why? Well, because now we know which genes are important for the life cycle, and we also know which genes appear to be variable across our donor population. By combining the two, ideally, we'd actually be able to identify which genes are contributing to that phenotypic variation we observed. So we found 7 genes that overlapped. We filtered so that the genes, the EQTLs, that had the strongest effect on gene expression rose to the top, and that's how we found IFIDM3. So what we're showing here is that donors with the CC genotype at this genomic site, which is denoted by the SNP identifier RS344811144, donors with the CC genotype have high expression of IFIDM3. Donors with the TT genotype have very, very low expression of this gene. Remember, this is an antiviral gene. So presumably you want a lot of this floating around your cells to protect you from these types of infections. But before we came to the hypothesis that CC donors may be more protected than TT donors, we first took a deep dive into literature to see what else is known about this variant. Previous work has found that this is actually located in the core promoter of IFIDM3, and that donors with the T allele at this site increase repressive factors at the CTCF domain, which is why you have lower expression of IFIDM3 if you have this base pair at this site. In addition, others have worked out the mechanism by which IFIDM3 protects us. So they found that this protein actually resides in the membrane of endosomes, and it can detect the presence of certain RNA, certain enveloped RNA viruses. Now, when it notices those viruses are present in the endosome, it actually shuttles that endosome to the lysosome for degradation. Okay? So if you have fewer copies of IFIDM3 floating around, fewer transcripts, fewer proteins, that sentinel system is not as strong, and you increase the probability that that virus, or the endosome that has that virus, is able to sneak through the system, uncoat, and get to the ER for replication. So that's what led us ultimately to this hypothesis that the donors with the C genotype are more protected from infection than the donors with the T genotype. So to directly test this, We went back to that same village I've been just talking about from the beginning. We threw either Zika virus or mock infection, so it's 2 wells in this case, same exact village in both. We waited about 2 days. We fact sorted based on expression of the Zika virus envelope protein. So it's our way of identifying who's infected and who's not. We then performed census sequencing on each of these fractions and calculated our abundance ratios. And as you can see, our hypothesis was supported. The donors with the CC genotype had a negative abundance ratio, meaning they had a greater representation in the negative fraction, the uninfected fraction, compared to the infected fraction. The opposite was true for the TT donors. They had a greater representation in the infected fraction compared to the uninfected fraction. Now again, to validate that this wasn't some sort of weird byproduct of the village, that this actually is something intrinsic to these cells, we then went back to that original data set that I showed you, 24 donors cultured separately. All we did was group by genotype at this SNP site. And as you can see again, donors with the CC genotype have much lower levels of infection compared to donors with the TT genotype. In this data set, 24 donors explain about 1/3 of the variation across those donors. We then took a completely different set of donor lines, so 36 lines not included in any of these experiments, repeated this infectivity assay in an arrayed format, and in this case, not only did the relationship hold true, but the genotype at this site could explain nearly 60% of the variation across the donors. We then ran a GWAS on this data set here, and the only 2 sites in the genome that reached genome-wide significance were the IFIDM3 promoter site I've been talking about the last few slides, as well as another SNP that's in high linkage disequilibrium with this site. So again, we're able to fine map a GWAS hit to one locus. Okay, so one of the things that I really take away from this, that I think is one of the most interesting aspects, has to do with the allele frequency of this RS3448114 SNP. So looking at the 1,000 Genomes Project, they looked at genomes across the entire globe, different ancestral populations. It's very interesting to me that the risk allele, the T allele, is pretty much non-existent in populations like Japan, Southeast Asia, Africa, to a certain degree, South America. But the risk allele frequency is dramatically higher in European populations. As you can see, in some cases, it's well above 50%. Now, some of you may notice that the map I'm showing here is color-coded red and blue. The red actually corresponds to the terrain inhabited by the A. aegypti mosquito, which is the vector for Zika, dengue, yellow fever, West Nile, and who knows however many ancient viruses that may have existed throughout the course of human history that are spread in these mosquito-laden zones. So this introduces a potentially interesting idea that perhaps the presence of these viral vectors has selected against the presence of the risk allele in these populations where the viruses are present, and the absence of that selective pressure has allowed the risk allele frequency to be much higher in these European populations. Now, I think this is interesting and potentially also clinically relevant because it's anticipated, it's predicted that over the next 30 to 40 years, because of climate change and increased globalization, the terrain inhabited by these mosquitoes is going to move further north, away from the equator. So it's going to be in places like Florida, Texas, and even here in Los Angeles. In Western Europe, it's predicted you'll see these mosquito vectors going up much further north and even approaching the Nordic countries at some point potentially, if you can imagine that. Now, there's 2 sides to this coin. One, that's quite scary because you could have a situation in which these populations are being exposed to these viruses for the first time without having this sort of innate protection from this protective/risk allele. But on the other side, perhaps we could toggle expression. Perhaps toggling expression of IFIDM3 could actually be used as a therapeutic, an antiviral drug target to help combat situations like this as they increase in frequency. I've been showing this slide for about 3 or 4 years now. Unfortunately, this is becoming all too true. Just last week, October 26th, California had its first case of locally acquired dengue. So dengue is now officially in California. It was in Pasadena, in fact. So if we think this is a future problem, it is not. It is a current problem that's only going to get worse. So brief summary of what I've talked about so far. Using NGN2 overexpression, we can convert stem cells to NPCs in a very, very fast and robust manner. We can use cell villages of these NPCs to capture genetic, molecular, and phenotypic heterogeneity in a shared in vitro environment. We can use dropulation to detect EQTLs in our villages, as well as using census sequencing to quantify phenotypic variation in our villages. I then give an example of how we can use this to identify, how we did use this, I should say, to identify a SNP in the IFIDM3 promoter that we believe plays a very important role in susceptibility to the Zika virus. Now, just the last couple minutes, I want to talk about the future of cell villages, primarily work being done here in my lab. I should have mentioned at the beginning, my lab is still in its infancy. We're 2 years old, so there's still a lot of work we've been doing to kind of get to the point where we're generating our own stories. As soon as we're done here, I'm going to rush back to the lab. We're building another village today for a really exciting experiment. We are in full village construction mode. In about 6 months, this talk might be very, very different because it'll be much more focused on what we've done here rather than what was done in my postdoc. So what I am most excited about for the village is its versatility. Okay, so we can make villages from any ancestral populations. In theory, it can be made from any cell type you can create in a dish. You can even, I don't even talk about what you can do outside of a dish. There's a lot of work being done using similar tools in which you don't even culture the cells. You say, like, take blood samples from a bunch of people, mix them together for these types of experiments. I could talk more about that in the 20-minute session at the end. But in theory, we can make any cell type you can make in a dish, we can make a village of. We can then also introduce various challenges beyond just Zika virus, right? We can use other viruses. We can do bacterial pathogens. We can add patterning factors to ask how genetic variation influences some of the earliest stages of human development. We can add a bunch of different toxicants and really challenge these cells and ask how do each donor respond differently to, say, valproic acid or ethanol or lead. We could even, in theory, perform drug screens in which you ask the question, how does a drug work in these different genetic backgrounds? This is really important to me because I'm Salvadoran, and there's a lot of studies showing how drugs don't work as well in minority populations, often because the preclinical work was done in cells of from European donors, and often the clinical trials are also skewed towards individuals who are from European descent. So the idea here could be, what if during the preclinical stage of drug development, they had to show that the drug worked in diverse populations? That's one thing you can do with a village in which it can be done in very, very high throughput. So what are we doing? Well, my lab is really interested, as I said right up front, in neurodevelopmental disorders. So one of our targets is trying to understand autism a bit better. One of the most common genetic causes of autism is a rearrangement known as 16p11.2 microdeletion. It causes this syndrome. It accounts for maybe 1% of all cases of autism, which is actually a very high percentage. I know it seems small, but autism is a very diverse disorder. The deletion of this segment increases your chance for not just autism, but ADHD, intellectual disability, seizures, and macrocephaly, or enlarged brain. So one of the nice things about this disorder is that the Simons Foundation, which is based in New York City, has done a lot of work to get patients and families together so they can generate a bunch of cellular resources from these individuals. And as a result, there are many cell lines available from patients. So we've already built a village of both patients and controls. We're not only looking at gene expression differences, but we're also looking at things like cell growth. Why? It's because, again, macrocephaly is associated with this disorder. Do we see increased growth in our neuroprogenitor cells from patients compared to controls? I'll tell you, early indications are yes, we very much are. Not only can we identify that phenotypic difference, but we can also look at the gene expression profiles and find associations between those two, right? Not just the genetic linkage, but also are there expression of different genes that are contributing to that differential phenotype. So that's one bucket of work. Another ongoing project is funded by the Simons Foundation, in which we're trying to address this really, really big question in the autism field, which is there's hundreds of different genes associated with this disorder, but we don't know how they all lead to the same set of symptoms, right? There's hundreds that do different things in the developing brain, yet they all lead to the same core symptoms. So what we're trying to do here is we're building a village that represents 11 different high confidence autism risk factors. So these are patient lines. We're pooling them with controls, and we're asking these same questions I presented in the last slide. How are their genotypes influence gene expression differences, as well as cellular phenotypes? And again, we're not just looking at these under static conditions. Mainly, we're not just asking, how do these cells differ from these cells when we culture them in a dish? The brain is not developing in a static vacuum, right? It's being constantly bombarded with different developmental cues. So we're modeling that as well. We're taking these villages, these patients and controls, we're throwing things on like sonic hedgehog agonists and Wnt agonists, these very, very powerful signaling factors. We're trying to stimulate these cells and ask, is perhaps the disease mechanism not in how the cells behave just under static conditions, but in the response to important developmental cues? Is that the key to understanding autism and these disorders in general? Now finally, a project that I'm really excited about that kind of came out of nowhere. In fact, I was giving a very similar talk about a year ago, and Patrick Allard came up to me and said, hey, I have some ideas for what we can do with the village as it pertains to neurotoxicity. And he said, basically, we do the same experiments we do with Zika, but we're going to throw lead on your cells instead. I'm like, great, it's very easy. We can use the exact same village and everything. And so what's interesting about lead, what's important, I should say, about lead, is that lead exposure early in life is a major contributor to neurodevelopmental disorders and other neurological issues. Recently, the World Health Organization has changed their guidelines. They used to say, like, this amount of lead is safe, and they lowered it. Every 5 years, they kept lowering it. Now they're saying, like, actually, there is no safe level of lead in your blood. If you have any lead in your blood, it can increase your risk of brain disorders. Currently, and this number is probably out of date because they really haven't looked at this more recently, about 500,000 people in the United States show elevated blood levels. 500,000 children, I should say. Globally, it's in the tens of millions. There's a study done just a few months ago that showed that 80% of kids in Missouri had detectable levels of lead in their blood. If you're wondering where it's coming from, you're like, well, wait, we got rid of lead from paint. We got rid of lead from our gasoline. Well, it's in batteries, and I hate to tell you, but it's in dark chocolate. There's been many studies coming out over multiple years saying that lead can be found in dark chocolate. We've actually stopped buying dark chocolate. And I remember when that study came out, we went to Ralph's, and all the dark chocolate powder had been piled up, and there's none of the milk chocolate left, because I think everybody read the same article, and so no one was buying dark chocolate anymore. So I highly suggest you try to avoid dark chocolate for a little bit. What's nice about the village, though, is it allows us to look at the gene-environment interaction, just like we did for Zika. And so one of the things that we generated for some preliminary data, this is some excellent work by Anna Vega in my lab during her rotation. She did this work where she took 12 different cell lines, or NPCs, and all she did was throw lead on them. This is an arrayed format, so it's not a village quite yet. And she asked over the course of 7 days, who dies, who doesn't die? Very simple question. And as you can see, some of our donors, similar to with Zika, right, some of them showed very little cell death. Others had 80% of their cells gone within a week. Okay, and this is normalized for differences in growth rates as well. So when I see something like this, it tells me this is ready for a village. So working with Patrick, we got some seed funding from the Broad Stem Cell Center here at UCLA to make a 100-donor village, throwing on lead, doing this sort of viability assays, as well as looking at gene expression profiles to ask ourselves, or to answer the question, what are the genetic and molecular risk factors for lead exposure? Are there people that are getting exposed to the same amount of lead, but showing vastly different vulnerabilities to that exposure? Hopefully, if all goes well, we will not only perform these experiments, but move on to other household neurotoxicants. Arsenic is a big one. Flame retardants are being shown more and more to be detrimental to early brain development, as are certain chemicals in cleaning solutions. I'm not trying to scare you, I'm just telling you what's being found. Okay, so thank you again for your time. I will acknowledge members of my lab. We started in 2021, as I mentioned before. Members of Kevin Eggen's group at Harvard, which the lab doesn't exist anymore, which is good for me because I now have no competition. He actually moved over to pharma. And then Steve McCarroll at Harvard Medical School, who is the brains behind, him and James Nemish were the brains behind the dropulation and census-seq algorithms. So with that, I'll take any questions. Yeah.